Hi, my name is Enoch David Johnson, and today I'll be talking about the vascularization of LSO in Fragile X model mice. Fragile X syndrome, FXS, is the most common single genetic cause of autism. FXS is caused by the FR1 gene being transcriptionally silenced, and this prevents the Fragile X mental retardation protein from being formed. Individuals with FXS have high sensitivity to sound. For this study, we use FMR1 knockout mice, and we use these knockout mice because they lack FMRP, just like how individuals with Fragile X syndrome also lack FMRP. And as a result, these knockout mice have similar symptoms to individuals with FXS. Knockout mice have an increase in excitatory and a decrease in inhibitory neurons in the brain, specifically in the LSO, which is a nucleus in the auditory brainstem. Moreover, we see there's significantly more astrocytes in the LSO of FMO1 knockout mice than in wild-type mice. So the current study was conducted to determine if the increase in astrocytes is associated with excessive vascularization within the LSO. So this means that since there's more astrocytes within knockout mice in the LSO, we want to see if this causes more blood vessels within the LSO also. For the methods, there are four main steps. So first, we took the FMO1 knockout mice in wild-type tissue, and we applied these onto slides. So we took P21 mice, and we took cross-sectional slices of their brains and applied them to slides. So here are the example of some slides we used, and O1 slides meaning wild-type mice, and O2 slides meaning knockout mice. Next, we use immunofluorescent labeling, and for the labeling, we wash the slides with PBS with Trinex with horse serum, and then we add a fluorescent antibody onto the slides so the blood vessels can glow, so we can analyze the data for microscopy and data analysis. And also, we use isolectin for the antibody to make the blood vessels to glow. Next, we use microscopy, and for the microscopy for each slide, and for which means each brain, we took four LSO images, and for each LSO, we took a 10x and a 20x image. So a 10x image allows us to count the number of blood vessels within the LSO, and for the 20x image allows us to calculate the percent of the blood vessel area to the total area of the LSO. And next we use data analysis, and for the data analysis, we use a software called ImageJ, which allows us to calculate the data. So for the data collection, here's a picture of a 10x image and a 20x image. So for the 10x image, it allows us to count the number of blood vessels within the LSO, and the shapes you see inside are the blood vessels within the LSO. And for the 20x image, it allows us to calculate the percent of the blood vessel area to the total area of the LSO. So for data analysis, this is for the 10x image, and we count the number of blood vessels within the LSO. And for the 20x image, allows us to calculate the percent of the blood vessel area to the total area of the LSO. And so for the results, this is for the 10x results, and we see for the wild type and knockout mice, for the wild type mice, the number of blood vessels within the LSO is around 75, while for the knockout mice is around 70 to 79 blood vessels. So as you can see, there's no significant difference between wild type mice and knockout mice for the blood vessels count. So for the 20x image, for the density of the blood vessels within the LSO for the wild type and knockout mice, we see that percent of the blood vessels in the LSO is around 5% for the wild type mice. And for the knockout mice, we see it's around 5.5. So again, we can see there's no significant difference for the density of blood vessels within the LSO for both wild type and knockout mice. To further determine the significance between wild type and knockout mice for the blood density, we do a ANOVA, which is a statistic technique to analyze if there's a significance between the data. And since the p-value for the ANOVA for the 10x image is above 0.05, we can see there's no significance between wild type and knockout mice for the blood vessel count in the LSO. And for the 20x image, again, we see the p-values greater than 0.05, so there's no significance between wild type and knockout mice for the blood vessel percent area in the LSO. So in conclusion, there's no significant difference between knockout and wild type mice for blood density / vascularization. The difference between the number of astrocytes in the FMR1 knockout mice is not a reaction to abnormal vascularization, but maybe it could be the result of abnormal development factors of cell signaling. So for a future study, we could see if abnormal development factors of cell signaling affects the number of astrocytes within the LSO. Thank you.